time to talk about this really cool thing. I'm going to start with the introduction. For those of you that have been here during the lecture series, I think this is the third time I've been on stage and the last time I'll be on stage. My name is Amy Glassmeyer, and I'm the department head of Urban Studies and Planning, which is over in Building 7. And you can guess I don't work on this subject. But I was interested in this because Bruce, Logan, and I used to be at Penn State. I came here a year ago, and we served on committees, and we were in the same room many times over, and I never actually got a chance to go over to his laboratory and find out what he was doing, but it just seemed interesting to me that he was making really things you'd squeeze between your fingers and would kind of be gummy, actually have the potential to generate energy. So I thought that the best thing I could do was to invite him here and let you see him and let me learn something about what he was doing. So he is is it the CAP or the? Cappy,
like happy?
Okay. Cappy Professor of Environmental Engineering in the Department of Civil and Environmental Engineering at the Pennsylvania State University. He has degrees from Rensselaer Polytechnic as well as his PhD at the University of California, Berkeley. We share an alma mater. You didn't know that. He's had a meteoric career in the last 10 years and has all matter of appointments in organizations at his own institution, as well as a series of academic appointments in institutions around the world, such as he's a visiting professor with rights and privileges in the School of Civil and Engineering and Geosciences at Newcastle, visiting professor with rights and privileges in Harbin Institute of Technology in China, a KAUST investigator at the King Abdullah University of Science and Technology, which itself is a whole interesting experience, which maybe somebody will ask him a question about. And so he basically serves around the world in a number of important roles, and I know that because I have a colleague who works with him who says, haven't seen Bruce in a while. So that says something about what he's done. His resume was going to print out at 50 pages, and I decided for the sake of the environment I wouldn't do it, but all I can tell you is that he is remarkably well published. He runs a very exciting laboratory. I know his students love him. And his research is focused on bioenergy production for the development of an energy sustainable water infrastructure for both industrialized and developing countries. Using new technologies, it's possible to directly generate electricity using microbial fuel cells, those things I talked about, or hydrogen gas using microbial electrolysis cells from wastewaters and biomass. These systems have the potential not only to power the water infrastructure, but to produce net excess power for communities and industries. I was just at a meeting yesterday where we were looking at the first fully integrated wastewater treatment plant that would generate all of its energy and actually sell byproducts. And it's going to be developed in Pennsylvania, and it's about a $36 million project. So they told me it was the first one of its kind. And so that was really pretty interesting. So I just want to say something about this idea of making power from a bacteria. In 2009, Time magazine gave the electric microbe one of the 50 best, an award for being one of the best inventions of 2009. It was number 20 of 52. And it says, bacteria has always gotten a bad rap, but we should be thankful for one especially talented microbe, Geobacter, which has a tiny hair-like extensions called pili that is used to generate electricity from mud and wastewater. I'm sure I'm just blistering these words. A professor at UMass Amherst has engineered a strain of Geobacter that's 8 times as efficient as other strains at producing power. The next step, creating a Bacter-based fuel cell that can generate cheap, clean electricity. So let's see what Bruce has to say about that. Thank you. Welcome, Bruce.
Thank you. Is this microphone working?
It's on you.
Does it work?
Can everybody hear?
Can you hear me through the mic? Okay. Okay, great. Well, thanks for that nice introduction. I spent a lot of time thinking about water, and when you start thinking about meeting the water needs of people around the globe, you start thinking about how much energy it's going to cost. It takes a lot of energy to meet those water needs and to meet those sanitation needs. And if you start thinking about energy, you got to think about climate change, because as we consume more and more energy, we're accelerating the rate of climate change. And of course, if climate is changing, then water distribution is changing. And if water distribution is changing, that may change requirement for energy as well. To put that in a different way, 1 billion people lack adequate water. 2 billion people lack adequate sanitation. And it's getting worse. By 2025, a quarter of the world population can be in severely water-stressed areas. Climate change is altering these patterns of runoff and water transport, and so the kinds of things we have today are going to be changing in the future. So it's just a little global map of surface temperature changes. Everything's getting warmer pretty much in the picture. Not too much interesting happening in the eastern U.S., but in the western U.S., it gets a lot more interesting because not only is it getting warmer, but as shown by these orange areas, there's a decreasing amount of water runoff that's going to be occurring in these areas. So what that tells us is it's going to be warmer, it's going to be drier, there's going to be less warmer, and there's probably going to be more people. We already may be seeing some of those changes occurring. This is Lake Mead in Arizona, where you can see the carbonate precipitation on this being a nice little historical record of how much the water level has dropped in that lake. Now, the energy that we use for our water infrastructure is about 3 to 5% maybe of all the electricity we make. That's about 21,000 megawatt power plants, just going for our water infrastructure. And the energy that we're using for our water infrastructure is increasing. We're seeing a lot more membrane processes being used, and increasingly people are turning to desalination as the competition for potable water increases. Also within the wastewater field, we've moved from much simpler treatment processes like trickling filters to more energy-intensive activated sludge systems to membrane bioreactors. So at the very same time that we know we have less and less energy that's going to be available or as much more competed for, we're using more and more energy in this infrastructure. But let's turn that around. If you actually look at wastewaters that are produced, there's a lot of organic matter in those wastewaters. And right now we're using energy to get that energy out. What if instead we extracted that energy? Well, we'd have almost as much energy as equal to the electrical energy that we now use to run our water infrastructure. Now, I'm not saying that we can get 100% of the energy out, but I'm trying to cast that number in light of proportions. And in fact, at any wastewater treatment plant, we almost have 10 times more energy in the wastewater than we use to treat it. So we shouldn't be throwing this energy away, and we ought to be thinking about how to make our water infrastructure sustainable, because I think we'd rather be using that energy for other things. And so my own goal is really to try and make this happen in the next 20 to 25 years. Come meet me in 20 to 25 years and we'll see if I made that. How do we do this? Well, we're going to need some new ideas. We're going to have to learn to think outside the box, try and come up with new ways to create energy, right? Okay, we'll talk about that in a minute. After I try and entertain you for a few minutes with a few fun slides, I'll talk about what this technology is really about, and I'll spend a little time talking about the microorganisms that help make this possible, and then I'll leave you with sort of a sense of where things are going in terms of transforming this into a practical system outside the laboratory. Here's one approach I like for thinking about new ways to think about water. It's called virtual water, where you look at how much water it takes to make a product, and therefore you can either decrease the amount of water it takes or choose another product. So a cup of coffee uses 140 liters of water from over its whole production time. Well, if you look at a glass of beer, that's only 75 liters. So I think what I'm saying is you should get up in the morning and have a glass of beer, and you'll save some water. Although actually I think it'll make you thirstier. Or you could get up in the morning and have some tea. Well, that's good. That's making better use of a resource. But we really need to come up with new ways of making energy. And so the first thing you do is you say, let's look into nature and see if there are ideas in nature that we can adapt. Or here's another approach. Let's use our imagination and let's try and think about new ideas we could come up with. So I thought, well, let's think about what they did in all these science fiction shows and movies. How did they have enough energy to do all that? They were zipping around space. So I went back to the good old standard in in science fiction, Star Trek back in the 60s. And what did these guys have for power? Anybody know? Dilithium crystals. Look at that. There you go. And has anybody ever seen a dilithium crystal? I haven't seen one. So until one comes along, I don't think we're going to have any answers from Star Trek. But, you know, I don't think they really had that many answers because look at this cell phone they've got. I mean, the thing's big. It doesn't have a camera. And it's got no keyboard. You can't even text. I mean, what were they thinking of? A little bit further into the future, back to the future, we had Mr. Fusion, right? But, well, not quite. Actually, this was an ethanol-powered vehicle. You could see that's a can of Miller, I think, there. And then maybe a little bit further, there's the Matrix. Now, this is a future where you can wear any color clothing you want as long as it's black, to paraphrase Henry Ford. What did they do in the Matrix? Well, what it was really about was that they had taken humans and plugged them in to generate and extract electricity. So really, our idea, this new idea, would be to plug in people at night and extract electricity from people. Well, there's not that much electricity in people, and I don't think we want to be doing that. So we thought, well, instead of using people, why don't we wire up bacteria and create these sort of biobatteries, or as nature called it, circuits of slime. I think I like biobatteries better. But this is a microbial fuel cell that's in the lab. This is a fuel cell that contains nothing but bacteria and water and something for the bacteria to eat. The bacteria break down the organic matter, they generate electrical current, and that does useful work, in this case running a fan, and I assure you there is no battery inside that box. So this has led us to create 3 different technologies so far. One we call microbial fuel cell that makes electricity, one that we harvest the current that the bacteria are generating to produce hydrogen or methane, and the third one is what we call a microbial desalination cell. So I want to tell you about each of these for a little bit, and then I'll tell you about the bacteria that make them work. So this is how it works. The bacteria eat organic matter. They break down that organic matter, release electrons. There's no oxygen for those electrons to flow to, so the electrons, but some bacteria can release these electrons to an electrode. They flow to the counter electrode because there's potential difference. And at the counter electrode, oxygen is reduced to water with the electrons and the protons, and that maintains the system. For every electron that's made, there has to be a counter ion produced so that charge is balanced. So this was one of the very first systems that we made, produced less than up to, but when we first started doing this, like a milliwatt per square meter of surface area. So we immediately tried to make these systems produce more power to understand what was interfering with or what was limiting power generation. And I want to give you, I know that you're not technically active in this area, most of you, so I want to just give you a sense of kind of how the apparatus has evolved and how we're making these things. But also what really makes this particular field interesting is that you all can be inventors. This is the kind of thing where students in middle school are making these and winning science fair projects left and right. There's no book on this. Anything goes. This is a whole new technology. So there's a lot of room for a lot of inventorship. So here's the first thing we did. We got rid of the cathode sitting in water because it takes a lot of energy to put oxygen into water. So we used an air cathode like you would in a hydrogen fuel cell. And just by going to that air cathode and moving the electrons a little bit closer, we got a 1 to 2 order of magnitude jump in power production per surface area. Now, when I say maximum power, here's what I mean. There's a voltage produced by across the circuit. What we do is we change the external resistor or the load on the surface. When we change the resistor, we change the voltage. From the voltage we produced and the resistor, we can calculate the current. From current and voltage, we have power. And then we produce what's called a polarization curve, where we show what happens to voltage as a function of current And most importantly to our discussion is this power density curve. This is how much power we produce. So when I say power, power is really a function of the current. I mean the maximum power right up here, the top of what this system can do. That's how we're going to use our yardstick to compare the performance of this system. So take this reactor with that air cathode. It had a piece of carbon paper here that the bacteria grew on. The cathode was another piece of carbon paper. In this case, it had a catalyst for oxygen reduction. We covered that with a proton exchange membrane because every single fuel cell that we'd ever looked at had a proton exchange membrane. But when we started to think about it a little bit more, we realized that we didn't need a proton exchange membrane. The water was the proton exchange material. And so we pulled the membrane out, and it worked actually a lot better. However, it did leak water now. The membrane was holding the water in, so we had to put a coating on there to prevent the water from leaking out. We call that a diffusion layer. And it also helped to reduce the amount of oxygen leaking in that the bacteria might use. And we used an ammonia treatment process to make the bacteria adhere to the surface and to improve the current densities. And with those changes, we went from a half a watt per square meter now to 2 watts per square meter in power. Now we tried to continue to provide more real estate, if you will, for the bacteria to provide increased surface area. So we're sitting in the lab, you know, we're cleaning some test tubes. Well, not really. We're cleaning some beakers, you know, and we're holding this thing, this bottle brush up, and we're going, I wonder how we can create a high surface area electrode that is very porous. And the answer is right in front of us. This is made of graphite fibers, which are electrically conductive. And then this stem here actually serves as a current collector to collect all these electrons into a more efficient electron transfer material. And this graphite brush electrode has up to, it can have as much surface area as you want, but this one here has about 15,000 square meters per cubic meter of volume. Very high surface area per volume and very easy to make. We took that brush anode and put it in one of our reactors, and we also replaced the platinum catalyst with a cobalt-based compound because we don't want to use a precious metal in this system. And without any precious metals, and with these materials, we were now producing about 2.5 watts per square meter, and now starting to think about volumetric densities, how many watts we'll get per volume of reactor. We have to, like we increase the surface area of the anodes, we have to increase the surface area of the cathodes in order to pack enough electrode surface area into the system. So we think the answer to doing that is to use tubular cathodes because we can put a lot of these into the system. So we basically need to figure out how to make tubular electrodes that will provide us an air space on the middle, in the center, and then the interface for the water around the tube, and then make it electrically conductive so the electrons can get there. And we have some hope, at least for turning a tank into a battery, that this will work because in wastewater treatment we use these membrane bioreactors that are cassettes of these tubes. So intrinsically we have the technology to make a lot of tubes and insert them into water. We just needed to make them electrically conductive. So initially we tried a variety of materials. This is just carbon paper which is rolled up and coated so it doesn't leak, and these are 2 tube cathodes. And there's a brush anode in the middle, and this is just a brush anode in the middle, and actually the whole thing wrapped around with one big cathode. And what we found was that in all these systems, you know, it's not the bacteria that was really limiting the power that we got. It was the surface area of the cathodes. As we increased the surface area, it didn't matter whether it was this shape or it was flat or anything else. It was the surface area of the cathodes. So these cathodes are key to making these systems to work better. And they also cost a lot of money. There's another component to this, which is to, as we make these larger and larger, to do a good job, an efficient job of getting the current through the circuit from the anode all the way over to the cathode. So we've been looking at using current collectors which are stainless steel mesh. Stainless steel is very inexpensive. We can make any size mesh that we want. And to sort of build our electrodes around current collectors. The anodes already have a current collector, but the cathodes hadn't had one of these current collectors. So this was a new idea to take the stainless steel and now to build the electrode around the current collector. So we put a conductive paint right onto the stainless steel mesh, we found that this is our mesh in blue, power density curve for our mesh. This is one for carbon, paper, and platinum. And you can see these things produce about the same amount of power, but we didn't need any precious metals. And now we have an efficient platform for scaling these up because we built in the current collector into the system. But there's another way that we've been thinking about, and that's using activated carbon as a cathode. Activated carbon has a tremendous amount of surface area per volume, and it also turns out that it is catalytically active for oxygen reduction. And so what we've been looking at is trying to make cathodes out of something that is really, dirt cheap. I mean, they make these things out of coconut shells and pits and that sort of thing. So this is a cathode that was manufactured by a company in Belgium called Vito. It's activated carbon. It's got no platinum on it whatsoever. And here's our Vito, the activated carbon cathode. This is, if we put platinum on it, it works about the same as our carbon cloth. And then this was the other one I showed you as well. So we're all right in about the same range. The platinum ones work better, but it would be nice not to have to use platinum. The other aspect of this is not just the materials, but the electrode spacing. And if you've done any work in batteries and so forth, you know that you want to try and get your electrodes very closely spaced. Well, that's a good goal, but the problem is if you get them too close, the oxygen leaks in through the cathode and the bacteria don't produce any power. They just use the oxygen. So you need what's called a separator, something to kind of cut down on that oxygen transport in. And once you do that, you can make the system very compact, pack a lot of electrodes per volume, and with a separator of cloth, a group in Oregon was able to get the same amount of power per area, but now they were looking at a kilowatt per cubic meter because of the density of the electrodes per volume. Unfortunately, the cloth that they used, this is like a handy wipe that you use to clean the kitchen sink. It's very porous. Biofilm grows on it, but once the biofilm grows on it, it eats it up. So actually, their separator got used as fuel. But we've been doing some work with glass fiber materials, non-biodegradable materials, and these work just about as well as the J-cloth does. So we think that this will help bring those electrodes together. So that's sort of a snapshot of the development of MFCs and the different types of things. And you'll notice none of those inventions required any manipulation of bacteria. Nothing about the bacteria. And we went into this, we thought it was all going to be about the bacteria. We were wrong. It was all the chemistry and the physics. When I got into this around 2002, and probably the first guy that did this was Byung-Hun Kim in Korea, and they were working down at 1,000th of a milliwatt per square meter. And just over the past 10 years or so, we've seen literally 5 to 6 orders of magnitude of power increases in these systems. Now, I don't think we can continue that trend. This is not going to be Moore's law. We are starting to see where we're really starting to maximum out on this. From a mass transfer point of view of substrate to a surface in water with some assumptions of bacterial kinetics, I estimate there's probably a maximum of only about 15 to 20 watts per square meter before we run into some diffusional limitations and kinetic limitations. There's already been some work done showing up to 7 watts per square meter when we use a really big cathode compared to the anode. That is, we get rid of that cathodic limitation. And typically, we're down here around a few watts per square meter for equally sized electrodes. Volumetric power densities, you know, this is not going to be the kind of thing you put on the back of the car in Back to the Future. These volumes they carry around a lot of water. You know, typically we're getting something on the order of a few hundred watts per cubic meter. Records up to a couple of kilowatts per cubic meter. And, you know, so what does this translate to volumetric power densities? Well, if we got 1,000 watts per square meter, that's not right. That should be a watt per square meter. We got a watt per square meter, and we got 100. Sorry, if we got 1 to 10 watts per square meter, we got 100 square meters per cubic meter. You know, we're still looking really at a kilowatt per cubic meter. I don't think we'll do much better than that. So this isn't going to be something that is really transportable power, but in a fixed location, it could translate into productive power. The costs, if I were to build just 1 cubic meter cell a few years ago, it would have cost me, you know, $250,000. Now we're looking at materials costs of a few $1,000s. And just to put this in perspective, a wastewater treatment type of system like a trickling filter costs about $500 per cubic meter of material. So we're getting down into the range where at least this could be applicable in wastewater situations. Let me show you kind of the power or how the wastewater treatment, as I mentioned, how a wastewater treatment plant might be turned into a power plant. This is a power plant. This is a wastewater treatment plant for 100,000 people. Coming into this is 2,600 kilowatts, or the equivalent of, or 2.6 megawatts. Of that, roughly a third goes in for treatment, in direct and indirectly. The solids come down here and go into anaerobic digesters for methane. About 100 kilowatts is used just in this one process, and overall in the plant about 300 kilowatts. If we recovered this methane efficiently, we could have a treatment plant that would run itself. Not so far-fetched to have a treatment plant meet its own energy needs, but let's take that activated sludge unit out and let's put in a microbial fuel cell. And now with the methane and the microbial fuel cell, we might be looking at something like, take out that anaerobic digester and make a fermenter, close that loop. We might get up to something around a megawatt of power produced at that treatment plant. Sorry, skip that one. So what that means is we've gone from a treatment plant that was consuming roughly a third of a megawatt of electrical energy. That's like a megawatt of primary energy, right? A third of efficiency. Now it's producing a megawatt, which means we no longer have to consume 3 megawatts of energy to make that electricity. We're talking about a change of about a 4 megawatt balance of primary energy from a net consumer to a net producer or replacer. Okay, let's switch gears now and talk about microbial electrolysis cells. And this is the same sort of platform, but since this is an energy series, let me talk energy for a minute. You take glucose, cellulose, look at its heat of combustion, how many kilojoules we have per mole. And if we were to turn 1 mole of glucose into 2 moles of ethanol, we might have just a little bit less combustion energy. And if we were to turn stoichiometrically 1 mole of glucose into 12 hydrogens, we'd have 3,400 kilojoules, which is great because that's more energy than we started with, which of course you know you can't do, which just tells you thermodynamically you got to put energy into that system if you're going to get 12 moles stoichiometrically out. A gallon of gasoline is equal energy-wise to a kilogram of hydrogen. But if you were to look at building a fuel cell car and not producing, say, a gas guzzler, you wouldn't need so much hydrogen to run that car. You might only, say, need about estimates of 4 to 5 kilograms of hydrogen for the same range that maybe 30 gallons would take you in a well-loaded SUV. So we don't have to use as much hydrogen, and it's not quite so far-fetched to imagine that we could run a fleet. And I'll come back to this point in just a little bit. But the problem is that most of the hydrogen we make is made from fossil fuels. So, you know, have we really changed the playing field if we just turn fossil fuels into hydrogen? Well, a lot of people don't know that bacteria will ferment cellulose and make hydrogen. While theoretically you could get 12 moles of hydrogen per mole of glucose, in nature you can only get 4. And that's if the bacterium produces only acetate as its product in the liquid. You could get 4 moles of hydrogen. In practice, most reactors produce somewhere around 2 to 3 moles of hydrogen per mole of glucose. So there's this fermentation barrier, and it's really a thermodynamic barrier. You can't go uphill. And so the question became, well, how can we get past this? How can we make more hydrogen? Because I really want to drive one of those hydrogen cars. So here's what we came up with. You take a microbial fuel cell, which is producing electricity, and then you don't give it any oxygen, you take it away. Well, then nothing happens. It's an uphill reaction. But if you boost the voltage that's produced by the bacteria with an external power source or with a microbial fuel cell, you now have enough energy in that system to drive current production, and at the cathode you get hydrogen gas evolution from 2 H pluses, 2 electrons, gives you 1 hydrogen. So it's a way of electrochemically making hydrogen from the voltage and the current produced by the bacteria. Theoretically, you only need about a couple tenths of a volt for this organic matter electrolysis using bacteria versus about almost 2 volts are used in practice for water electrolysis. So I won't go through in the same detail that I did with microbial fuel cells, but suffice it to say that we've been looking at trying to make these systems more efficient, learning from what we learned in MFC design. This is a brush anode. This was, in our first steps, a platinized cathode. There's no membrane, short electrode spacing. And we got, compared with acetate, which is the breakdown product of cellulose or glucose, we got 80 to 95% stoichiometric recovery as hydrogen of what we could theoretically get in this system. And we got energy efficiencies over 100%. Isn't that great? You always want to break thermodynamics, get over 100%. Well, it's manipulating things a little bit. This is the electrical energy input. So compared to the electrical energy input, we're getting about 2 to 4 times more energy back as hydrogen. When you figure in the substrate energy, of course, you've got to be below 100%. But still, we're looking at 60 to 90% energy efficiencies. We also tried to make a high surface area cathode, and we've been looking at non-precious metals for this as well. This is actually a cathode made out of stainless steel, which in phosphate buffer actually is very catalytically active for hydrogen evolution. Tried to kind of merge these two technologies together, sort of half hemispheres to keep the electrodes close together but maintain the high surface area. And this is a series of cycles of current generation. And you can see our stainless steel brushes performed as well as our earlier designs with platinum and carbon cloth, which both are quite expensive. Okay, that brings me to the third technology, which we call a microbial desalination cell. I was on a plane. Amy said I travel a lot. I was on a plane coming back from Saudi Arabia. I'm thinking, you know, I've really always wanted to come up with a new way to desalinate water. And I'm reading this review paper on membranes, and I say, my God, I think I can use a microbial fuel cell to do this. And I started making all my notes on the plane, you know, and I get home and well, 2 weeks later I'm in China, and I find out they've come up with the same idea 2 months before I did. So we decided to collaborate. Now here's how this works. It's based on using the properties of membranes that selectively pass charge. An anion exchange passes anions, but not cations. Cation exchange doesn't pass anions and does pass cations. So take a microbial fuel cell and put in two membranes. Got the bacteria over here eating up the organic matter, and when they do that, they release a proton. Well, the proton can't pass the anion exchange membrane, but charge has to be balanced. And so a negative charge moves out of that central chamber, which contains my salt water that I want to desalinate. So as the bacteria produce electricity and release protons, the chloride's removed from the center chamber. The electron goes over to the other side, combines with a proton in the water and oxygen, but a proton is removed on the other side. Therefore, sodium has to come out to balance the loss of that charge. And as a result of those two processes, the sodium chloride in the center chamber is desalinated. We haven't used any electrical energy off the grid, and we have produced power. I can't tell you too much more about that. We only kind of discovered we could do this. We're working on improving it, but let's talk about the bacteria that can do this. I call this, actually, I think Yuri Gorby first coined this word, electromicrobiology, which I think is kind of a nice way of saying it. I see this as a sort of a new subdiscipline, a new sort of opening our eyes that these bacteria are capable of this electron transfer. But so far I've talked about electrogens, that is, bacteria that can generate this current outside the cell, take the electrons from inside the cell to outside the cell. Well, you have to figure if there are electrogens out there, that there ought to be something that's accepting those electrons. We call those electrotrophs. So this is not well agreed upon, but let me tell you about how we think things are working. So bacteria, you put them in one of these fuel cells, they rapidly colonize the electrode, they produce these biofilms, which look like they're multicellular, multi-layer biofilms that are functioning to produce that current. Right? They're just working just like you and I. We're breaking down organic matter. Those electrons go through a series of respiratory enzymes. In those respiratory enzymes, we produce fuel for ourselves, ATP. The bacteria do the same thing. And so respiration is really where the energy is captured. It's not in the eating, it's in the breathing and digestion. And so we think that the bacteria that are functionally important in this area, many of them are actually derived from bacteria that can respire with solid metal oxides. And let's see if this works. Okay, that wasn't encouraging. This is a little film put together by Ken Nielsen and his group out at UCLA. When you live in Hollywood and you study bacteria, you make movies about bacteria. So that's what these guys did. And these are Shewanella oneidensis. They're bacteria that are eating lactate. And they can use oxygen to breathe, but they can also use solid metal oxides. This is a particle of manganese oxide. The bacteria eat the lactate that's in solution, and then when they breathe, they release the electron to that solid particle of manganese oxide. When they do that, the manganese becomes soluble and dissolves away. So they are literally breathing away this solid particle of manganese oxide. And in doing so, gaining energy for themselves and also killing themselves, because when that's gone, they have nothing left to breathe. Here we go. So we think that these microbes are important, and we also think that one of the mechanisms, and this is of the mechanisms that's most fascinating, that the bacteria are producing electrically conductive appendages. And this is sort of like the wired and the wireless internet. You know, if you need to release these electrons, you might as well wire up to where they're going, and you can transfer them more efficiently. The evidence for nanowires is primarily based upon looking at conductive probe microscopy, where they drag an AFM tip over one of these appendages, and you can see the ones that Geobacter make are maybe you can't see, but these are very thin. These are individual, we believe, individual nanowires. This looks more like sort of a cable of nanowires. And as you probe over the top of this, you get, you can see that this thing is electrically conductive. So it's like an uninsulated wire. So we think these microbial communities are these nanowire producing communities. Other bacteria that produce electron shuttles that move through from the bacteria to the surface, some that need to make direct contact. And then wherever you have a biofilm, you always have bacteria living off the other bacteria. What kind of bacteria can do this? Well, it's not as if it's one kind. You know, we went into these biofilms and people looked at it. There were all these different kinds of bacteria under the categories of alpha, beta, gamma, delta, proteobacteria. And we said, is this possible? Are all these guys really making electricity? And so we put them into pure culture tests and then looked in pure culture if these bacteria could do this. And this is a pretty extensive list. So it's not like there's one bacterium out there that can do this. There's lots of them. And we don't know why. And there are probably different reasons, not just to respire with iron or manganese. But it's clear that the bacteria that can respire with iron and manganese produce good power. This is just a mixed culture, an environmental inoculum. This is a pure culture of Geobacter. Produces just a little bit less power in this system than the mixed consortium. But does the ability to respire with an anode necessarily require metal reduction? And the answer is no. This bacterium can respire with iron, but it can't use an anode. And there are several bacteria that have been isolated that can't reduce iron, but can use an electrode. This Rhodopseudomonas palustris is an interesting microbe because it's being studied for its ability to make hydrogen. It turns out it's also capable of producing high power densities. In fact, this one produced more power than the culture it was derived from. Another bacterium didn't produce much power, but it was interesting because not only could it make electrical power, but it could also do it using just cellulose. So one microbe that could break down cellulose and make electrical current. We find unexpected guests, and we find superheroes, hidden superheroes. So this is a community analysis, and we see these Geobacter, Rhodopseudomonas I just pointed out, and some other ones we expect. But a lot of times we see uncultured. We don't know what these are. We got to find them. We go into something which is fed acetic acid. We expect to find nothing but Geobacter sulforeducens. But our community shows us we have all these pelobacters. These are they uninvited guests? What are they doing there? Other substrates. Oh, look at this. We see Geobacter, but Geobacter is not exactly known to use succinic acid and iron. What's it doing there then? We have these things that we run sometimes for years. We see these strains evolve in here. We don't know why they're there. We do transfers. They don't go away. In this case, this Paracoccus was producing just as much power as the mixed culture. So it's evidently responsible for power generation. But we went in and we found another isolate, Shewanella, and it produced more power than the Paracoccus denitrificans. So why didn't it take over? There's obviously other ecological benefits for being in these systems. Okay, let me talk about the other side of this, electrotrophs. When we have electrodes in MFCs, on the cathode we can get microbes that catalyze the reaction. They can use, for example, nitrate as an electron. They take the electron from the electrode and send it off to nitrate. Or they send it off to oxygen. But what if these microbes are out there functioning in our MFCs, they ought to be fulfilling some sort of role in nature. This is a shot of a fermentative organisms, breaks down organic matter, and it's connected with this appendage to a methanogen. Well, this appendage, it turns out, is electrically conductive. And so the hypothesis is that this microbe is wired up to that one. directly transferring the electrons from a substrate of the oxidized to the methanogen. So this is interspecies electron. So this is a variation of what's called interspecies transfer. An interspecies hydrogen transfer, the prevailing theory is that one microbe breaks this down, makes hydrogen. That happens the electrons move very fast through this. Then you have the slow diffusion of hydrogen over to the methanogen, and then another fast process. But why have that slow process? Why have that dirt road in the middle of two highways? Why not have the electrogen get together with maybe an electrotroph and connect these up, and then have direct electron transfer? So this is what we think is going on in nature. And we think that these electrogens are making these appendages, and they're hooking up with the electrotroph, but we don't really understand this process. Is it like only one microbe hooks up to another microbe? Is it, this one has this kind of plug and that one has that kind of socket? Or what about the microbes that live in England? They got different different plugs over there. Or maybe they're in Europe and the plugs look like that. How specific is all this? I mean, but maybe there are universal sockets that say, well, we don't care what this is. We'll just adapt to any old key. And maybe there are microbes that aren't very choosy and they sort of have universal plugs too. So understanding how specific this is, what that reaction is, what that relationship is going to be a lot of fun to figure out. But with this, we could take a source of electrical energy and put these methanogens to turn that electrical energy into methane. And to do this electrochemically uses a lot of precious metals. These methanogens can do this, and we've shown that we can generate methane with these methanogens on a cathode. So the idea is to take an MEC and instead of making hydrogen, make methanogens on the add methanogens on the cathode and make methane in this way. And that's not going to displace how we already make methane, which is in these large digesters, but maybe this would be a new way to turn a source of electricity into a transportable fuel. Okay, so let me finish up now and tell you just a few things about scale-up. Savings. If we can do this, turn wastewater into something that makes us money, great. If we don't need to put energy into the system, even better. And if we don't need to make all those bacteria, we make a lot of bacteria when we treat wastewater. It's called sludge. We wouldn't make so many bacteria because we're extracting that energy out now. We'd save the operation of the plant a lot of money. First larger scale MFC was done in Queensland, Australia. These were individual microbial fuel cells. The water came up through the middle and flowed over the outside. They're each 3 meters tall. Worked okay, didn't generate much electricity. It's a project going on in Connecticut where they're turning wastewater into electrical energy. These are working okay. They're somewhat cathodically limited. And that place I was coming back from, we hope to actually build one of these maybe starting this summer, although that timeline's slipping. But we were able to build a larger scale MEC to make hydrogen. And we got some funding. It takes a fair amount of money to do this, so we got some funding from a few different sources. We evaluated some different wastewaters. We looked at a domestic wastewater, human wastewater, from a winery, from a dairy, and this was a place that processed potatoes into potato chips. So where would you go in these 4 places if you could go somewhere to set up a pilot scale reactor? You're right, we chose the winery. It turned out the potato chip waste actually works better than the winery, but we had already decided that a winery in California seemed like a really good idea. So here's San Francisco, here's the winery up there, there's the tasting room. They didn't we didn't go in there too much, but the idea is we just pull the wastewater off their existing sump, send it into an equalization tank, and then into our reactor. This is what the thing looks like. It looks like a big kind of refrigerator or bathtub, depending on your point of view. The wastewater actually goes around the back and comes in from this side, but goes around the back and then flows back through the system. We have a gas trap there. Remember, we're trying to make hydrogen, gas collection and measuring, and we have a water seal because hydrogen is very leaky We want to try and collect it and not have it leak out through the crevices. This is the back end of the tank where you can see that flow line coming in. It's actually the little tube there. The bigger tube is just a quick fill and empty tube. We can run it in recirculation mode. We have sampling ports, but the interesting part is up there where we have the electrical connections, you know, and of course anybody knows that, you know, toasters and water don't go together, but here we are, you know, we've got electrical energy in a big tub of water. This is the water flowing through the system. Each one of these is one module. There are 24 modules. And each one of those has a total of 6 pairs of electrodes. We have a plastic mesh for support. On top of the mesh, we have a glass fiber separator to make sure the electrodes don't touch each other. We have a brush anode and a stainless steel mesh cathode, so it's just a piece of stainless steel screen, if you will. And a current strip. This is how we collect the electrical current from the anode and send it over to the cathode. And these things are this was really easy to make. This looks like a sheet of lighting grid. You throw down your cathodes, flip it over, put on your anodes, screw them together and pop them into the tank. You make an electrical connection across this collector strip. That collector strip comes to the edge of a tank. We put a bolt through there that goes to the outside and hooks up to those wires that you saw on the outside. And put all the modules in, cover it up. This is our electronics shed. We've got a pump controller for flow into the system, a series of power supplies. Each module had its own power supply because we wanted to look at how the different modules did in performance, a multimeter for our measurements, computer for data storage, and our prediction of performance. Here's how much hydrogen we should have been able to make. This is our laboratory data with this little reactor. We figured we would scale based on cathode surface area. So we looked at how much cathode surface area we had in this thing. We scaled that over to current, and we predicted we'd be running at about 9 amps for this cubic meter reactor. So we took the thing out, and we started it up on an auspicious date, as it turned out, on September 11th. And we predicted 9 amps, and well, we didn't get there. Not much happening here. We tried to get some more. We were just using the raw wastewater. We got some solids out of the lagoon. We added in some activated sludge solids, and tried some other things. Still didn't. We started now to see a little bit more current increasing. It took us actually about a couple months to get up to, we finally got up to around 7.5 amps. And then we got tremendous rainstorms in California, flooded the sumps, we had a water heater to keep it warm because it was getting pretty cold in Napa at that time. And the heater broke and the water flooded and everything jumped down, and we got it fixed. And right now we're still running the reactor. But we made close to the current that we thought. The problem actually was that we ended up making a lot of methane. We couldn't bubble the current out fast enough, and the methanogens were consuming it. So we needed to increase the rate that we got the system working. About out of time, right? Is that good? Okay, so let me wrap up. Good. Let me leave you with a few other thoughts. I have talked a little bit heavily about wastewater because I think this is probably the first place this will be implemented. And because I am interested in all that energy we use for our water infrastructure. But you can certainly use a lot of different things in these fuel cells. Cellulose, chitin, you can grow algae. Some people want to grow algae and make a diesel fuel. You can grow algae and have them just throw them in as a source of organic matter. You can use side products from other types of fuel production. But consider just cellulose for a minute. If you take cellulose and you put it in a microbial electrolysis cell, you can make hydrogen at pretty good yields, 8 moles per hexose equivalent, but the rate is very low. So if we split that process up into first a fermentation step where in this case Clostridium thermocellum ferments the cellulose into hydrogen and volatile acids like acetate and so forth. then we need a much smaller reactor because it's much more efficient to work with these materials than with the cellulose. And we're able to maximize our yields a lot better too. This system was running at about 10 moles of hydrogen per hexose equivalent. The theoretical yield is, limit is 12. If we were to take hydrogen, make hydrogen from this cellulose, how much hydrogen do we need? And can we make, and do we have enough cellulose? So a study was performed that showed that if we started to transition to a hydrogen infrastructure, that by, this isn't going to happen overnight. Gas cars are still going to be there, but we'd increase the amount of hydrogen vehicles. We'd eventually need something on the order of about 10 to the 11th kilograms per year of hydrogen to fuel all light-duty vehicles in the US. The DOE has estimated that we have available to us, if we work at it, about 1.4 billion tons of cellulose. If we turn that cellulose into hydrogen at stoichiometric yield, we'd have 2 times 10 to the 11th kilograms of hydrogen. If we do it at 50% yield, we'd have to have that. Well, we're in the ballpark. So this tells us that there's theoretically enough cellulose out there to fuel our light-duty vehicles in the US if we want to do that with it. Okay. That concludes my talk. I just want to say that I hope I've introduced you to a new field, convinced you that there's some interesting invention and engineering to be done out there. But we've seen that really in a relatively short amount of time and with not that many people in the world working on this, we've increased power by several orders of magnitude. We've substantially reduced the cost of these materials. And along the way, we've sort of discovered a whole new field of microbiology. We need to do more advancement of materials. We need to understand the microbial communities better. And we need to be more aggressive about some of these pilot plant studies, which are going to take money, but we think that there's great return on that investment. A lot of the work I talked about today was done by my group. This is my current research group, and they're really very excited about this work, and they do great work, and I'm just pleased I have a chance to present what they've been accomplishing in the lab. If you want to find out more about this, you can go to my webpage, or if you like the old-fashioned approach, you can buy the book, or there's an international website. And my son, he said, "Dad, you need a Twitter account." I said, "What's Twitter?" You can see there's a little bit of a generational gap there. And so he explained it to me, and so I even have a Twitter account now, although I can't say I send many messages out. And there's a webcam here. You can do this on your phone or your computer or whatever. Hopefully it's running while I'm standing here, but we've run these webcams where we have that fan spinning in real time, and we've run those for anywhere from a year to a year and a half before we take it apart and kind of put in new parts and newer technology and get it running. And with that, if we have time, I'd be happy to answer any questions. Thank you. Yeah, so the question is maintenance. You know, the one thing about technology that you've only just developed is you only know how long it's going to last by running it, for a long time. We do find that the cathode builds up a little bit of a biofilm, and that's probably our greatest concern. The anode actually seems to continue working just fine because the bacteria at the surface are alive and breathing, and as they get pushed away through new cell production, they stop breathing, and so they kind of fall off. So it's sort of self-sustaining at the anode. At the cathode, we may need to work hard to keep that clean. And so the answer is I don't know, just because we keep changing the material so fast, we don't have a good sense of that. But certainly we don't want these things to work a day or a week, and ones like that MFC fan have run at least a year, year and a half. Yeah.
If you take a MEC, if you take an MEC and you have a certain rate of hydrogen production per unit area per second, and then you channel that hydrogen into a non-microbial fuel cell with hydrogen and oxygen, do you do better than 20 watts per square meter? In other words, rather than doing the microbial fuel cell, which is doing it all with microbes,
Yeah.
you have a half microbial and a half non-microbial approach to building a fuel cell. Do you actually do better than 20 watts per meter squared? Because if the hydrogen production is slow,
Yeah.
are you still limited?
Well, I don't think I can simply answer that. If you're mating an MFC, for example, to an MEC, the energy recovery in the MFC is not as good as the energy recovery in an MEC. And in the end, I couldn't tell you, I couldn't make that calculation with any confidence. But I think, no, I'll just leave it at that. And I'd rather have a piece of paper and a pencil in front of me before I went through that one. Yeah. In the back there? Yeah.
I was wondering what the reasons for most of these MFCs being cathode limited are. Is there some biological reason why anode reduction is more efficient or happens faster than cathodic oxidation?
I think at least on the anode side, the bacteria are really just limited by their respiration rates. At the cathode side, we're more limited by our understanding of how to catalyze oxygen reduction. And we're working in an extremely difficult regime of pH, which is circumneutral, temperature, room temperature. And under those conditions, platinum just doesn't even work that well. So the bacteria are really good at pumping out electrons and protons. We just have to get the protons from there all the way over to here. That's a slow step. We have to catalyze that reaction, and we're not doing a good enough job at that. So I'd just say, nature has figured out the anode part pretty well. We got to figure out the cathode part. Was one over here?
Going along the same lines as pH.
Yeah.
Going along the same lines with pH, in your desalination fuel cell, it looks like you're building up a fairly high pH quickly, right where the bacteria are living. Is that going to be a limit to the efficiency of that fuel cell?
Yeah. So in the desalination cell, the cells produce a lot of protons, which is actually a low pH, but a high high proton concentration. And they are acidifying themselves. And that is a limitation. You can put buffer in to ameliorate that, but look at the other side of it. You're consuming protons. So if you can recycle your anode solution over to your cathode, you essentially could be balancing out that proton flow. So that's our hope anyway. There's some other things we're toying with that might be a little bit more efficient, but I can't tell you those. But there are ways to get around that. But you're right, that's a really big stumbling block. If they, in a poorly buffered situation, it's just going to stop things in a hurry. I think there was one. Well, there's this guy with the hand up, and now, okay, there we go. I can't see him, he's in the light. Yeah.
So is there any investigation of optimal scale for such fuel cell assemblies? Say, would it be better off operating in a distributed system, as in distributed power generation, or would it be more economical or efficient to operate at large-scale central stations?
So the question is about scale. Should we build a lot of little MFCs or a few big MFCs, right? Yeah, and the easy answer is we haven't even figured out how to build a really good, MFC to be able to say how big should we make it. But the expectation is that no matter what scale we try and go to, these will be modular in nature. That is, we won't try and build one the size of this room. We'll try and build 10, that are 1/10 the size of the room. Because when you get too large in distances, you've got to send electrons from over there all the way to somewhere else and make this thing into one huge MFC. That seems more difficult to do than to build lots of small modules that you can just crank out and add on or subtract off as you need. But I do expect that at one central location it would be easier to handle the power you generate and so forth. But I'm guessing. I don't know. I mean, I don't think anybody's really been able to think that far ahead because we haven't achieved an efficient prototype yet.
We're still at the lab scale.
Well, we're still getting out of the lab kind of thing. Once we start building these multiple ones, then we'll be able to answer that next scale question.
So the industrial scale.
Yeah. There was a question over here. Yeah.
Yeah, all right. So most of your talk was focused on fuel cell architecture and anode surface area, just as you said yourself. So in most of the cases, using something like genetic engineering to knock out a bacteria's ability to produce pyrite would kill current density. But in the particular case of that interspecies hydrogen transfer, you said it yourself, if there was a way to knock out that pyrite production, that dirt road between the two highways, you force the bacteria to touch each other, you might be able to increase methane at a faster rate. So I know you didn't touch on anything about the microbiology aspect of it, but do you know any groups that are, instead of targeting fuel cell architecture, using synthetic biology or genetic engineering?
Yeah, so really the question is, can we genetically engineer these microbes, whether it's at the anode or the cathode, to work better for us? Ultimately, yes. There are some groups that are trying this. Derek Lovely's been working on genetic engineering of Geobacter, but to date, nobody's done better than just either using a good strain or the way they increased the current density for Geobacter was they left it on the electrode for half a year at a poised potential, and it adapted to that condition. So there was no genetic engineering. Nobody that I know has figured out a way yet to genetically engineer the organisms to increase power. And that's primarily because we're still not completely sure about how it all works.
Well, let's give our guests
Oh.
an applause.
Okay.
Is there another question? Okay.
No.
Last question.
Sorry.
I imagine biological systems are finicky in nature.
Yeah.
And that small changes in the environment can greatly impact the microbial consortia.
Uh-huh.
I was wondering if you could say something about the standard deviation that you've seen in similarly built MFCs.
Yeah, so variability from cell to cell. I'd say probably sometimes 100%, as in it died. But that's a rare occasion. Usually we start these up and inoculate two side-by-side cells and less than 10% variation amongst those cells. So really, things gravitate very quickly towards the electrical energy production and in very reproducible ways. We have one reactor we keep in a temperature-controlled room. We keep running that. And we want to start up a new one, we just go in and pour out that into our reactor and it takes right off. So we don't generally see variability, but we can create variability. And as we create that variability, we'll understand, we can understand why it is, and that'll be the fun part. So, and that is the last question then, right? Thank you.
There's a reception in the back for the first one to get there. So run like a microbe.
Right.
So I actually had some questions about the microbial populations themselves.
Yeah.
So when you showed this pilot plant in Napa, you showed that you changed the different wastewaters that you're adding in.